We've talked about Mendel a lot, but now I want to get into non-Mendelian genetics. Complex inheritance. Complex inheritance. So when scientists discovered Gregor Mendel's work on the basics of genetics, it was hailed as a major breakthrough. For the first time, scientists could reliably predict and describe what was happening when two organisms with known traits were bred to produce offspring. Scientists began noticing that not all traits that they predicted in test crosses followed Mendel's laws of inheritance. One example of this is eye color. Scientists determined that although Mendel was correct in his ideas, the big picture of genetics is a lot more complicated. So, incomplete dominance. So, the dominant allele is not always completely expressed when the recessive allele is around. So the phenotype will be somewhere in between, like paint mixed together. So an example of this is in snapdragons. So a red flower is capital R, is going to be crossed with a white flower, which is little r. This cross will form pink flowers, which would be big R, little r. So incomplete dominance is when you have the two alleles, but one is not completely dominant over the other. So let's do a practice problem with this. So if tail length in cats is an incompletely dominant trait, what would be the resulting offspring in a cross of a long-tailed cat, which is big H, with a short-tailed cat, which is little h? So here are our genotypes. So if we get big H, the cat will have a long tail. Little h, the cat will have a short tail. But if we get big H, the cat will have a medium tail. So we are crossing a long-tailed cat with a short-tailed cat. So the answer is that 100% of the offspring will have medium tail length because all of our boxes have a big H and a little h because we brought our big H's down and our small h's to the side. Let's do another one. Flower color in orchids is an incompletely dominant trait where homozygous individuals are blue with big H, or yellow, little h in color. So in a cross between two green orchids, what percent of the offspring would be expected to have green flowers, blue flowers, or yellow flowers? So let's keep in mind that big H would mean the flowers would be blue. Little h means the flowers would be yellow. And big H means the flowers would be green. So we are crossing two green flowers. So we have big H and little h on both sides. Remember, anything on the top you're going to bring down. Anything from the side you're going to bring over. All right. So our answer is that 50% will have green flowers because we've got two big H and little h here. 25% will have blue flowers since we have two big H's here. And 25% will have yellow flowers, signified here. Now let's talk about codominance. This is when the alleles work together. So both traits are going to be fully and separately expressed. So when an organism has two different alleles, for example, if it is heterozygote for something, it'll express both at the same time. And an example that we see this in is roan cows. So red cattle are homozygous dominant with big R, and white is also homozygous with big W. So when they are crossed, they will produce a red and white baby cow. So the difference between codominance and incomplete dominance is that incomplete dominance, you're mixing colors just like you would in paint. So you can't necessarily see the red and you can't necessarily see the white because it comes out pink. But with this, we have a white parent and we have a red parent. And you can see both colors are fully expressed on the baby cow. So let's do a practice problem. So in certain fish, blue scales, big B, and red scales, little b, are co-dominant. So when a fish has the hybrid genotype, it has a patchwork of blue and red scales. So cross a patchwork fish and a red fish and determine the genotypic and phenotypic ratio. So we know that big B is going to equal blue scales, little b is going to equal red scales, and big B, little b is going to be our patchwork of a mixture between blue and red scales. So we are crossing a patchwork fish and we are crossing a red fish. So we got our patchwork fish up here with the big B and little b, and we got our red fish, little b. So remember, anything on the top comes down, anything on the side comes to the side. So our genotypic ratio will be 1:1 because we've got 2 big B, little b, and we got 2 little b. And while 2:2 is not necessarily incorrect, but it is not the most correct answer. So we simplify that down to 1:1. So the phenotypic ratio is going to be 1:1 of patchwork to red scales. Let's do another one. Now, in some chickens, the gene for feather color is controlled by codominance. The allele for black feathers is capital B, and the allele for white feathers is capital W. And the heterozygous phenotype, speckled, is called erminant. If 2 erminant chickens are crossed, what is the chance that the offspring will be white, black, or speckled? So, this is when our genotypes start looking a little strange. So, big B, or we've got our H's here. So, we got our genotype up on the corner. That'll equal a black chicken. So, big W, or HW is going to be white. And then BW, or an H with a B, H with a W. This is our speckled chicken. Now, the reason we write it like this is so you don't get confused with the different letters. It makes it a little easier if you're working with the same thing, and then you just have your letters up here kind of as a coefficient. So here is what it'll look like if we are crossing 2 speckled chickens. So 25% of the chickens will have white feathers, 25% will have black feathers, and 50% will be speckled. Now let's talk about polygenic traits. So a trait produced by 2 or more genes, it shows a range in phenotype. So an example of this is skin color, height, IQ, and eye color. So genes from multiple chromosomes can be used to code for a specific trait. So in this diagram, part of chromosomes 3, 5, and 8 are going to come together to produce the proteins needed to code for that trait. Now with multiple alleles, which is having more than 2 alleles for one gene, an example of this is rabbit fur. So imagine your big C, this is full color dominant to all other alleles. So now we got our little c with our CH coefficient. This is chinchilla. So it has a partial defect in pigmentation. So it is dominant to our little c and our C with a coefficient of H alleles. Next, we have our little c and our H. So this is Himalayan. So Himalayan color in certain parts of the body are dominant to the little c allele. And the little c allele is the albino no color, which is recessive to all other traits. So our key matches up with our rabbits over here. So here are the possible genotypes that we can have for our furry friends with multiple alleles. So let's do a practice problem. So let's cross a full-colored rabbit that carries the chinchilla allele with a chinchilla rabbit that carries the Himalayan allele. And what is the phenotypic ratio of the offspring? So here we have our full rabbit, but it carries that chinchilla allele. So our full rabbit is represented by the big C, and our chinchilla allele is represented by our C with our CH. So now let's cross it with a chinchilla rabbit that carries the Himalayan allele. So here we got our chinchilla and then our Himalayan allele. So let's cross it. All right. So your genotypic ratio is going to be 1 to 1 to 1 to 1 because we have every single box here looks different. But your phenotypic ratio is 1 to 1 because we have our full color to our chinchilla. So these two are going to be represented by your full colored. Well, these two are represented by chinchilla because they don't have that full colored capital C dominance. So let's do another one. So let's cross a Himalayan rabbit who carries the albino trait with a chinchilla rabbit who carries the albino trait. And what is the genotypic ratio of the offspring? So let's cross a Himalayan rabbit that carries the albino gene. So we are going to have our Himalayan and the albino gene. And here's our chinchilla and the albino gene. So let's cross this. So once again, our genotypic ratio is going to be 1 to 1 to 1 to 1 because all of our boxes are different. But you will have a 2 to 1 to 1. So you'll have, for every 2 chinchilla, you'll have 1 Himalayan and you'll have 1 albino. Now, I'm sure you're wondering, wait a minute. We've got this. This comes first before this. So how does that count as the chinchilla? So it's 2 chinchilla. Just because it is written here, it is still dominant over that CH. Even though it's in the second spot, it still dominates, just like it does here. Now, another example of multiple alleles is blood type. And there are 4 different blood types. You have A, B, AB, and O. Now, what determines your blood type? Blood type is determined by proteins or antigens on your red blood cell. So, for example, type A blood contains the A antigens on its red blood cells. Now, blood type consists of 2 dominant allele options and 1 recessive allele option. So A and B are dominant, and O is recessive. And A and B show codominance. So here are your blood type alleles. So the way that we write this is capital I can show that you are full type A, or you can have a capital I and a little I, which shows A and O. These little I's will represent O. So B is going to be written the same way as A. A and B can only have 1 combination of alleles, just like O. So let's do a practice problem with this. So let's cross a man with type A blood who is a carrier for O with a woman that has type O blood. So what is the genotypic and phenotypic ratio for the offspring? So we're going to put woman up top, man off to the side. Okay. So your genotypic ratio is going to be 1 to 1 with a phenotypic of A blood So A blood is going to be represented on this side, and O blood is going to be represented on this side. Let's do another problem. So a male with type AB blood is crossed with a female with type O blood. So what is the genotypic and phenotypic ratio of the offspring? So we got our AB blood on the top, and we got our type O to the side. Our genotypic ratio is going to be 1:1, with the phenotypic ratio 1:1 being A blood and B blood. Because even though we crossed it with an O, that A and B are going to dominate over the O blood. So just a quick recall with your sex-linked traits. Humans have 46 chromosomes. Chromosomes are made up of DNA and proteins, and they contain your genes. 2 of your 46 chromosomes are your sex chromosomes. So the sex chromosomes are called X and Y chromosomes. Everyone has an X chromosome though. So females just have XX and males have XY. Now, sex-linked traits are traits that are specifically on the sex chromosomes. So most sex-linked traits are on the X chromosome because it's larger and contains more genes. So an example of this is hemophilia and blind color. Those are sex-linked traits. Now, it is more common for males to have a sex-linked disorder like hemophilia. What do you think this is? Because females have 2 copies of the X chromosome and males have only 1 X chromosome, X-linked recessive diseases are more common among males than females. So let's do a practice problem with this. So a woman that is a carrier for color blindness is crossed with a normal male. So what is the chance that they will have a child that is colorblind? So, according to this, they have a 25% chance of having a child that is colorblind because even though the N is present here, we have this other X that dominates over that N. And so that child will only be a carrier of colorblindness, but will not be colorblind. So let's do another practice problem. Hemophilia is a recessive X-linked genetic disorder. Affected individuals' blood does not clot properly. So let's cross a male with hemophilia with a female who is neither affected nor a carrier of the disease. So what is the chance that they will produce offspring with hemophilia? They have a 0% chance of having a child with hemophilia because since mom is not a carrier or has the disease, she is going to dominate the genes, so her children will not have this disorder. Now, I know that was a lot and that's a bit more complicated than what we've been working with, but remember, if you have any questions, do not hesitate